The overall goal of this protocol is to define standardized approaches for the construction of simplified model plant ecosystems, called EcoFABs, that enable controlled, replicated laboratory experiments to investigate plant-microbe interactions that can be further tested in the field.
This method can be used to study plant-microbe interactions within controlled environments that allows us to gain detailed mechanistic insights into the functions of microbiomes to allow us to harness them for sustainable agriculture.
We have designed this first EcoFAB that employs widely available 3D printing technologies and sterilized plant growth chambers that enable both non-destructive high-resolution imaging and chemical analysis.
The EcoFAB device developed here is well suited for the analysis of plant microbe interactions because it incorporates analysis of root morphology and mass spectroscopic imaging.
Following the construction of EcoFAB molds using 3D printing according to the text protocol, in a disposable 1-liter container, mix 40 milliliters of siloxane elastomer base with different volumes of curing agent, depending on the experiment. Place the container in a vacuum chamber for at least 30 minutes to remove air bubbles from the elastomer mixture. Then remove the mixture and pour it into the assembled 3D printed plastic mold.
In order to generate a uniform